Thank you very much for the invitation. It's great to be here and see old friends. And my talk today was supposed to be about algorithmic challenges in complete genomics and metagenomics. And this is my outline. And I was trying very, very hard to feed this outline. There is a lot of territory to cover. And I was trying very hard to feed this outline into 60 minutes, standard 60 minutes talk. And then I learned, that it's actually 45 minutes. So there is no chance. I cannot simply like remove the last item in this outline because it's kind of cherry on the cake, right? So I have to dramatically cut everything. I added one more section, and that's it. But the title changes. This will be the first talk ever I give without any signs of algorithmic advancement. No algorithm in this talk. Only biological implications of complete genomics and metagenomics. So relax, it will be very, very smooth, simple, and please interrupt me in the middle of the talk if there are any questions. So you remember that, some of you remember, that 24 years ago, human genome was completed. But not quite. There were thousands of holes. And also these holes were in some of the most biomedically important regions of the genome that happened to be very difficult to assemble. And finally, 3 years ago, the first complete human genome was assembled. And it was based on the high-canoe algorithm developed by my former student Sergei Nurk, who is shown here. And from this, there was an explosion of complete genomics projects. In particular, currently, there is a human pan-genome project that will soon generate over 300 complete human genomes. Why people are investing a lot of money? It's not, these projects are not inexpensive. Why people are investing so much money into this project? One of the reasons is getting to the dark matter of the human genome that remain unassembled or remain poorly assembled or remain assembled in a single individual, human individual, not anymore, in the previous efforts. So let me give you, just as a preview and remains from my previous talk, let me give you a light introduction into genome assembly. Genome assembly started, there are two algorithmic paradigms in genome assembly: overlap graph approach and de Bruijn graph approach. And overlap graph approach was used to assemble the draft human genome 24 years ago. At that time, de Bruijn graph approach was already developed, but it was viewed exotic and completely impractical, theoretical kind of development. And then, around 2010, short-read sequencing by Illumina emerged. An overlap layout consensus, overlap approach disappeared overnight because it turned out that de Bruijn approach is able to handle this high volume of reads and assemble them much better than overlap approach, since basically the whole short read sequence is dominated by de Bruijn approach. And then, not that long time ago, long but inaccurate read emerged. And at that time, there was so-called error myth that de Bruijn approach works well for accurate reads, but falls apart for inaccurate read. And once again, at that time, overlap approach was used to assemble genomes. And then my student Mikhail Kolmogorov demonstrated that de Bruijn approach actually works well for inaccurate reads and developed fly and metafly tools. So that was kind of settled. But then there was next revolution, I think it was like 3-4 years ago, high-fi reads, long and accurate. if the Bruijn approach works so well for short reads and accurate reads, then it must work well for hi-fi, long and accurate reads, right? But my student, Sergey Nurk, who was raised on the idea of the Bruijn graph, still used overlap graph for assembling, for the first assembly of the human genome. And at the same time, my other student, Anton Bankevich, finally developed assembler for hi-fi reads. And next year, Mika Rautainen and Sergey, together with Sergey Nurk, developed Verka assembler for hi-fi reads, which is other De Bruijn assembler that is currently very popular for assembly. Anyway, two students, two different stories, and interestingly, when there was a Assembleton for T22 consortium, the conclusion was the overlap graph is ill-suited for assembling highly repetitive regions. But the whole T2T project is for assembling this highly repetitive regions. So this is the major focus of assembly. So let me then just explain what is the challenge here. Well, assembling of read, De Bruijn assembly, is so well-studied problem. In particular, space assembler, which is currently dominant assembler for short reads, is very well developed, with 1,000 of different applications for whatever variation, metagenome, viral genome, plasmid, whatever you can imagine. Why do we need a new assembler? It's the same kind of, just longer, slightly longer reads. I mean, 2 orders of magnitude longer, but anyway, here's the problem. So yes, we have great assembler like Spades for assembling short and accurate reads. It looks like should be easy to modify it for long and accurate reads and assemble hi-fi reads. So why do we need a new assembler? That will be the only algorithmic concept I'm describing in this talk, just to justify that it's not a walk in the park to assemble long genome. Reminder of what is the Bruijn graph of a genome first. Given a genome, let's consider all k minus mer in this genome. If k minus mer repeated twice, we just put a single node in the graph. And then H set will be all pairs of consecutive k-mers in the genome, or in other words, H k-mer, right? So genome is simply a walk in the graph, of course, in the Bruijn graph. And the next, but we are not assembling genomes. We are assembling reads, right? What is the De Bruijn graph of reads? Well, very simple. You view each read as a mini chromosome in your virtual genome. There are millions of such mini chromosomes, and you simply construct the Bruijn graph of this virtual genome with millions of chromosomes. Every chromosome is a read. Make sense? As long as reads contain all k-mers from the genome, it will be identical, the Bruijn graph. Make sense? Okay. In my class, I always emphasize that classical definition of the Bruijn graph is bad for you, because it doesn't allow your fantasy to fly, okay? We always use a different definition of De Bruijn, the same, but mentally different definition of De Bruijn graph. Here it is, because it allows you to deal with errors. Here it is, here's the definition. So let's transform our genome in a genome path of overlapping k-mers. K-mers, right? Or k minus 1. I don't remember. Anyway, this is transforming the graph into a genome path of overlapping k-mer, and then let's glue identical k-mers in this path. So GA and GA are identical k-mers. Let's glue them. We'll get something like this. If we glue AG, we get something like this. If we glue CA, we get something like this. If we glue AT, we get something like this. Now, close your eyes. I see some of you already close your eyes. Close your eyes and imagine that you do all these gluing operations at the same time. That's what we get. And it's exactly the same. But it allows you to think differently about this de Bruijn graph. OK? Anyway, here's why we need completely new set of ideas for constructing de Bruijn graph of large genome. Now, de Bruijn graph of large genome are so large that you cannot fit them into any imaginable memory. OK? You need to store all chemers. How can we what can we do? Well, here TA is a non-branching nodes. And if we simply combine all the non-branching nodes, we will get non-branching passes. For example, ATTAAG is a non-branching pass, right? So the right way is to simply collapse all non-branching passes in the graph into a single edge and label it appropriately. And this very simple transformation results in roughly k-fold compression of memory for de Bruijn graph. And as a result, the memory footprint doesn't depend on k. So it doesn't matter how large is k, it's roughly the same memory, and we fit everything in memory. Perfect. So when I give the problem in my class, construct condensed de Bruijn graphs, that's what people do. They start from genome, then construct de Bruijn graph, and then condense it into condensed de Bruijn graph. It doesn't work because you cannot construct de Bruijn graph. It's just too large. So the goal is, how can you construct condensed de Bruijn graph without constructing de Bruijn graph? It turns out to be non-trivial. This problem was actually solved almost 10 years ago by Liam Minkin, Son Pham, and Paul Medvedev, who got recomp of best paper award for this paper some time ago. But it's constructing de Bruijn graph of genome. We are facing a much harder problem, constructing de Bruijn graph of Fried, so with many, many complications there. And Anton Bankevich actually solves this problem. And what we came up with was crucial for constructing good assembly is actually the concept of multiplex de Bruijn graph. In multiplex de Bruijn graph, nodes have different sizes. Some nodes have size 500. Other nodes have size 1000. That's critically important for success. And so that was the key idea. And then the same idea was used in the Verka assembler by Mikhail Kolmogorov and Sergei Nurk that is currently widely used. OK, so the only thing that was needed at the first glance looks like almost trivial step, simply increasing k size by 2 orders of magnitude. Turns out it's a difficult algorithmic problem, but it was solved. Now, I don't have time to talk about all this development in this area, but I will talk briefly about implications. First, something that was completely missing from the first draft genome assembly 25 years ago, centromeres. Obviously, biomedically very important, linkage to various diseases, but completely missing because it's so repetitive, very difficult to assemble. And there was also a centromere puzzle. Why such functionally kind of it's the same function, is so variable across human population, across different chromosomes. And there was no tool when we started it with T2T consortium. There was no tools for analyzing centromere because there was no centromeres. So that was a large, many, many different studies. I don't have time to go into this. Another problem that emerged, immunoglobulin loci, arguably one of the most biomedically important regions of the human genome, that by the time of the T2T project was assembled only in 2 humans. So difficult to assemble. And There are now with accurate sequencing, and by the way, it's, for example, it's assembled by the time in macaque. But assembly is so unreliable, so you cannot really make judgment because it's very difficult area. You cannot trust the assembly. I will talk a little bit more about this studies in the last few years. And another issue, antibiotics discovery. So let's focus this talk only on the last two topics, antibiotic discovery and population-wide immunogenomics. Let's start from antibiotic discovery. Yuan, very expensive cancer drug, isolated from C-squared. There are ships hunting for C-squared, 1,000 kilograms, 1 gram of Yuan. It turned out that C-squared is not at fault. This compound, Yuan, is bioactive ingredient of a peptide produced one of the symbiotic bacteria living in C-squared. There are hundreds of them. How do you find which of these 100 bacteria generate this specific compound? And this is a question about a metagenomics route to antibiotic discovery, which did not exist when we kind of ventured into this area. It's very difficult. New antibiotics is a huge development, is a huge enterprise at the cost of $1 billion. Anyway, we are all raised on central dogma. DNA makes RNA, makes protein. But is it possible to assemble a protein from amino acid without any RNA and without ribosome? What do you think? Who knows how to do this? Is it possible? No, I see people saying it's not possible. It is possible. And each of you actually is somewhat familiar with this process. There is so-called non-ribosomal peptide synthesis. When a giant protein assembles amino acid one at a time into a short protein peptide without any RNA template and without any ribosome. And non-ribosomal peptides are some of the most successful pharmaceutical compounds. Antibiotics, immunosuppressor, antiviral agents, you name it. So there's huge area for pharmaceutical industry. That process was discovered by Mohamed Marahel 25 years ago. But how this giant protein knows which amino acids to assemble into a protein? Well, there is a non-ribosomal code. And in like fantastic work, Marahel actually figured out how this non-ribosomal code works. But it is less precise than genetic code. And that's the problem. So what are these magical proteins that assemble amino acids without any RNA? It's the largest protein known, and it's a giant molecular assembly line. They contain, let's say, here we assemble short peptide consisting of 10 amino acids. This synthetase contains 10 so-called adenylation domains, or A domain. Every domain knows which amino acid to select to add to a growing peptide chain. And as a result, after 10 steps, you have a linear peptide of length 10, and then there is typically cyclization process, and you have cyclic peptide. Why most of them are cyclic? Difficult to destroy. Okay? So is this process clear? So that's how they are produced. The question is, what is the code? How this A domain knows which amino acids to load? And basically, that's how it works. If you take various domains, and if you align them correctly, which is non-trivial already, then there are certain positions in these A domains that define what amino acid to load to the growing chain. For example, these are 8 positions, and if code is like this, if code is 8 amino acids, then certain amino acid is loaded, okay? And it's not just 20 amino acids, it's 200, includes many non-standard amino acids. Now, this is a fantastic machine learning problem for those of you who want to have a real adventure, because non-ribosomal code is still not as precise, not perfectly explored as genetic code. For example, it gives you probabilities that certain amino acid is loaded, but you actually don't know which one. And that's the challenge. Even having non-ribosomal synthetase, we don't exactly know which peptide it generates. So there is no immediate direct way to figure out which antibiotic. So now let me tell you about this metagenomic roadmap to antibiotic discovery. We started this project maybe 10 years ago with this collaboration with Peter Dorstein at UCSD. So it starts from looking for interesting samples. These days, many promising antibiotic comes from marine environment. And then what we were, usually people just do mass spectra. What we kind of advocating that we should do both DNA sequencing and mass spectra on these samples. Now, when we have DNA reads, we can assemble the genome, right? When we assemble the genome, there are programs that allows us to find non-ribosomal peptide synthetases, and then we can simply, based on this non-ribosomal peptide synthetases, and based on mass spectra, we can ask two questions. Given mass spectrum, usually people use mass spectra to decode peptides, to figure out amino acid sequence of peptide. So we can ask what antibiotic is encoded by this spectrum. And given non-ribosomal peptide synthetase, we can ask a question what antibiotic is encoded by this non-ribosomal peptide synthetase. Seemingly, there are two ways to discover antibiotics. Unfortunately, neither of this way is reliable. Okay? So because of limitation of our knowledge of non-ribosomal code and because limitations of our ability to decode mass spectra. So our idea was neither of this is reliable, but together they provide complementary information. And if we can piece these two puzzles together, maybe we can figure out what is this antibiotics. And there was a long string of papers where we were trying to build this bridge between mass spectrometry and metagenomics. And we actually built this bridge. But while we were building this bridge, another bridge collapsed. And this is this bridge that collapsed. Because, of course, we started this in the short read era. With this short read, we never can assemble complete genomes, right? And even fragmented genome, these non-ribosomal peptide synthetases are giant. They will be broken in the fragment assembly. And if they are broken, we cannot do anything. We need complete sequence of peptide. So let me just show you how fragment of non-ribosomal peptide synthetase look in a typical assembly. Here it is. Try to figure out what is going on here. So our bridge that we built for so many years basically didn't work because of limitation of short-read sequencing, because it breaks this non-ribosomal peptide synthetase. So, and we had a number of projects where we seem to be doing everything right, but in the end, due to this collapsing bridge, in short-read collapsing bridge, we were not very successful. And then, just 3-4 years ago, this long-read metagenomic emerged, and it was a complete revolution. In the very first project, we assembled over 400 complete or nearly complete bacterial genome, and 700 NRP synthetase. That was unheard of. And so that was amazing source of potentially new antibiotics. The problem is that even if you have an NRP synthetase, without mass spectra, you still, particularly in this case, like if you move to a kind of new environment like Amazon, there are new NRP synthetases, and genetic non-ribosomal code becomes particularly problematic, particularly error-prone. So it's very difficult to kind of move further. But at least there is a starting point now. And there is a good hope now, because there is clearly a way now to basically like SynBNP program to synthesize these antibiotics and move ahead with them. I won't go into more details on this because it is not the only topic I was going to cover. This is the last section of my talk on how complete genomics affects studies of our immune system. So new viruses constantly emerged. And our genome is just too short to fight all this arrays of new viruses, unexpected. Moreover, unexpected. Our genome never saw SARS, never saw HIV. How possibly we can find, I mean, we don't fight HIV well, but how possibly we can find all these diseases? Well, a baby introduction into just one of immunoglobulin loci called heavy chain immunoglobulin locus. In standard human genome, reference human genome, there is this just 1 megabase region that is critically important for our health. It consists of 55 V gene, 27 diversity gene, and 6 joining genes. And in the process called VDJ recombination, one of V gene selected, one D gene is selected, and one J gene is selected. And they're fused together to form a new gene that serve as a basis for antibiotics production. Make sense? This is not splicing. This is a completely different mechanism specific for immune system. And that's how antibodies are formed. Now, important thing is that these genes have certain specializations. For example, most flu-fighting antibodies are derived from a single V gene called V169. You can also derive it from other. There are 55 V genes. But the chances, the probability that functional anti-flu antibody will be derived from other V gene is very small. It's mainly one V gene. Now, there are 14 known variants of this V gene in human. And roughly half of them have phenylalanine and position 55, and after half of them have leucine position in position 55. Why do I care? Well, because successful or unsuccessful binding to the flu surface protein will depend on how lucky you are with respect to position 55. And 15% of US population carry ineffective alleles, which means that you probably should not go for vaccination in this case. Okay? Now, there are many cases like this. So these V genes are specialized to find certain diseases. Now, you probably at this point, you should interrupt me and tell me, well, if there are only 55, 27, and 6 genes, there are only that many, hundreds of possible antibodies, thousands, right? We cannot possibly find all array of possible viruses. So how we still do this? Well, that's the whole story unfold. Thank you. You hear me now? Okay, good. So there is this process of maturation of naive antibody into functional antibody. Many rounds, but if you are lucky, and let's say within a week of this evolutionary process, naive antibody is transformed into antibodies that actually can find the virus, you survive. But if it is 2 weeks, you probably will be dead. So it's very important how this process goes. So with certain probability, you have a chance to develop functional antibody and naive antibody. What is special from the evolutionary perspective? What is special about antibodies then? With normal genes, you just need to have a mutation or not mutation to fight a disease. In this case, you have a different mechanism. You can simply, for example, if you are lucky to have a mutation that transform amino acid position 50 in V1-2 derived antibody, there is neutralization of HIV. But probability that you reach this state is small. And there is very few human individuals that naturally have this mutation. Okay? Now, what is special about antibodies? There is a different way to fight disease. You simply double the expression level of this specific antibody, and your probability of developing successful response doubles right away. But how do you double? Our genome found a way. And it will be, what I will describe is this amazing plasticity of our immunoglobulin loss, birth and death of immune gene. Now, if you look at immunoglobulin loci, it's like nightmare for Lyman or whatever. It's very complex. There are many highly repetitive regions that are difficult to analyze. And when we started, there were no algorithm to accurately align IG locus across mammals. So let me describe the challenges. Standard gene prediction methods are not applicable to immunoglobulin loci. So we had to develop gene prediction algorithm for loci. Standard aligners do not work for highly repetitive regions. That's what Human Pangenome Consortium wrote in their very first paper. More work is needed to determine how to align and represent large repeats array within pangenome. So we had to develop uniliner algorithm. Now, evolution of IG loci is manifested as a progression of duplication, but inference of duplications in highly repetitive genome is actually a very difficult problem. We had to develop genome decoder. And also, how do you compare highly variable duplication scenarios across multiple genomes? Well, it's something that we call differential comparison of segmental duplication. And this is also part of genome decoder. Now, what is the challenge in alignment? Let me just illustrate it this way. If you have ancestor here on the top, here, and it consisting of 5 units of repeats, and it produced 2 descendants. Each of these descendants is a 4-unit repeat. You start comparing S and T, and you see perfect alignment, nearly perfect alignment with just 2 mutations on your left, right? But it's completely wrong, because the correct comparison is right here. So you need to develop new alignment tool that actually allows you to find this evolutionary scenario, because otherwise you can't study evolution. Unless you establish orthologous immunoglobulin genes, you cannot say anything about evolution, and it has been traditionally a very difficult problem. In other difficult problems, there was shortage of immunoglobulin loci. They're so difficult to assemble, but finally, a few months ago, the primate T2T consortium assembled the complete genome of these primates, and that's provided very rich data for us to analyze within working with T2T, primate T2T consortium. So this is how dot plot of immunoglobulin locus in orangutan and human look like. You see, this region here shows that there is a region that was repeated in human 1 time, in orangutan 6 times, PPP. But you only kind of, here it's clear, but there are many other regions. And basically, since IG loci are shaped by segmental duplication, it is very, you have to decompose them into elementary duplicons, elementary blocks. You need to develop a new alphabet. But you cannot develop new alphabet unless you develop alignment tool for this, for analyzing this region. So block decomposition is not a new problem. There were many studies in the past that basically take 2 genomes in billions of nucleotides and develop them into something like this, where each block represents a duplication. But when we started working with primate T2 consortium, we figured out that neither of them is working right for extremely duplicated regions. And we had to develop a new block decomposition algorithm for duplicated regions on steroid like immunoglobulin loci. And here, take a look at the result. If you compare immunoglobulin loci in orangutan and human, there is like enormous evolutionary innovation within just 6 million years, 7 million years, right? Take a look at this. 6-fold increase in the block H, which means that each gene within this block has 6 times more usage, expression, which means that these blocks are now have better chance to fight infections which they are specialized for in fighting. Remember, we were telling that expression level or usage, people use usage in this case, is very important. So every duplication affects certain regions that are responsible for fighting certain diseases, and we clearly see death of genes, like here. This gene appears in human, doesn't appear in orangutan. Apparently, orangutan doesn't care about this particular, doesn't need to have V gene responsible for fighting this disease, or birth of new orange gene in human. And many, many cases like this. Now, I'm running a little bit out of time, so let me skip this slide and just tell you that I'm about to introduce disruptive structural IG evolution hypothesis, which basically statements that duplication represents a major mode of immunoglobulin evolution that enable a rapid response to new infection, unexpected infection. So let's discuss it. So the success of antibody response to a specific antigen often depends, as we saw, on a specific IG gene or V gene, doesn't matter. For example, response to malaria almost exclusively mediated by V333 gene. The usage of IG gene for us is simply a fraction of antibody derived from this gene in the whole antibody repertoire. Okay? So kind of an analog of expression, but it will be kind of something from 0 to 100. By the way, if some of your V gene have very high expression, very high usage, it usually means sign of disease. Now, duplication immediately affects the success rate of the antibody response to a specific gene, right? Because it increases the chance that following up somatic hypermutations, this evolutionary process, generate a functional antibody. Remember, we were describing this process. So it's not only mutations, but simply affecting which allow me to make a prediction then that high usage of anti-malaria V333 is more crucial for survival in sub-Saharan Africa than in the US. However, if you increase the usage of V333, unavoidably you decrease usage of other genes, right? So it's a difficult balancing game, how to define this use, to presumably optimize for each location based on spectrum of pathogen prevalent there. But remember, some pathogens are still emerging, so we don't even know which pathogen are show up. Now, I use this spectacular term simply because last month there was a paper that came out that used this term. Like, people are amazed what they see, the amount of variation they see within population. They use this term spectacular divergence for based on primer T2T results. So we already saw the standard duplication that hugely increased usage of some genes in orangutan compared to human. But the same thing is happening in human versus human. Human versus human is also very, very fluid. For example, here, K block increased. There are 3 K blocks, and there are multiple genes there. So what it means? Humans are constantly affected by newly emerged pathogen, which needs specific IgG gene, right? Implication is it is thus beneficial to have, for us, to have highly variable immunoglobulin loci, so that in this room, maybe there is at least one person that have V gene designed to find an infection that may be coming tomorrow, okay? And this is not kind of, Dobzhansky, almost 100 years ago, basically postulated that evolution promotes diversity, and the term disruptive evolution referred to the situation when an extreme phenotype has a fitness advantage over average phenotype. And this is also known for Ig genes at single nucleotide level, okay? What we see now with this primer T2T consortium finished, we see this at structural duplicon level with a different mechanism. So it's structural evolution. That probably explains why our Ig loci are so variable at the structural level. So a duplication usually doubles the usage of genes located in the duplicated block, and frequent duplication is an evolutionary mechanism ensure that population includes unusual individuals capable of mounting antibody response against new pathogen. It doesn't necessarily optimize the chances of mice survival, but it, because I may have suboptimal Ig gene usage with respect to newly incoming infection. Instead, it optimizes the survival chances of the population. And essentially, our immune system are like these Darwin finches. But it has not yet optimized their kind of morphology toward what they already saw. It optimizes morphology, it optimizes variation with something that is still unseen. Now, it's kind of wild hypothesis. How can we possibly prove it? Well, the Human Pangenome Consortium will soon generate over 300 complete human genome over various populations. An analysis of IgH loss may provide a way to confirm or reject the disruptive structural evolution hypothesis. We will go to sub-Saharan Africa and see whether certain genes, whether V133 or V333 is more duplicated in sub-Saharan Africa than in the US. Right? Potentially, there is a way to check it. Now, I'm almost on time, just 2 minutes late. These are great students in my lab who contributed to these various studies. And this is our collaborators at USDA and Wageningen University. Thank you for listening, and apologies that I haven't presented actually what I was planning to present, but there was no chance. But if you're interested, some of you know Bioinformatics Algorithm book that is now in 3rd edition. We thought about preparing the 4th edition, because it was about what happened in bioinformatics in the last 5-10 years. But then we realized the book will be over 1,000 pages long, so we now think it will be just additional book, almost ready. And I know that some of you are actually teaching from the book on the left. So connect with me, and we will send you this book that is almost ready. That's it.